Welcome to this video on genetic code, key features, structure and importance. After the discovery of DNA as the genetic material, a major question arose. How does DNA regulate biological functions? Watson and Crick proposed that DNA encodes its instructions in the form of messenger RNA, which is then translated into protein by translation. In this video, I will explore the central dogma of molecular biology, the genetic code, its key features, structure, and significance. So let us discuss central dogma of molecular biology. It explains how genetic information flows inside a cell. According to this dogma, DNA encodes the information in the form of messenger RNA through transcription. The messenger RNA is then decoded or translated into a protein which after modification performs biological function. In addition, DNA transmits information from one cell to another through a process called replication. RT or reverse transcription, which converts RNA into DNA, is also part of the central dogma. How the four bases in messenger RNA are used to specify amino acid sequence in a protein was predicted by Watson and Crick, which I will explain in the next slide. Let us define genetic code. What is a genetic code? There are four bases in a DNA or messenger RNA that encodes for 22 amino acids in a polypeptide chain. The question was, in what format are these four bases used to specify an amino acid? Watson Crick predicted that a group of three bases out of four are used to specify an amino acid. Therefore, they predicted that there are total 64 genetic codes to specify 22 amino acids in biological system. Regarding the definition and the size of the genetic code, a group of three bases specifying an amino acid is known as the genetic code. Later it was proven experimentally that there are 64 genetic codes. We will discuss the features of these code word or codon or genetic codes in the upcoming slides. So features of the genetic code. There are total 64 genetic codes which are shown on top of each amino acid listed here, 10 up and 10 down here. So there are total 64 genetic code which have been experimentally proven, each having a group of three bases. The first genetic code is AUG. This is also known as a start codon because the translation starts on AUG. There are three genetic code which are known as stop codon or nonsense codon or terminator because they stop they stop translation. They stop translation because there is no tRNA to read these genetic codes. However, the newly discovered amino acids such as selenocysteine and pyrrolysine are respectively encoded by the stop codon UGA and UAG. So there are three stop codons. The remaining 61 genetic codes are known as sense codon. Sense codon means they specify amino acids. So the next property, degeneracy of the genetic code. Methionine and tryptophan are the only amino acid which have only a single genetic code. Remaining all other amino acids, they have more than one genetic code, which is called as degeneracy of the genetic code. The degeneracy occurs at the third base. If you see in each group, the third base is variable. It means that the first two bases must base pair according to the base pairing rule to specify amino acid correctly. The base pairing at the third base could be non-standard. This is known as wobble base. So this is basically known as degeneracy of genetic code. Although there are more than one genetic code for most of the amino acid, but they are unambiguous. They in each group they all will specify the same amino acid. For example, valine has four genetic codes, but each will specify unambiguously valine in this case. So what is the significance of degeneracy? Without degeneracy, 42 nonsense codons would be expected. However, the degeneracy has reduced the number of nonsense codons to only 3. Therefore, degeneracy reduces the chances of mutation by allowing a tRNA to read more than one genetic code. Next property of genetic code is non-overlapping. What it means is that a genetic code once read is not reread partially or fully during a translation cycle. This allows the synthesis of an authentic polypeptide chain from a given open reading frame. So the next property of genetic code is that genetic code is not universal. It is almost universal. The reason is there are differences between the standard genetic code and the mitochondrial genetic code, as well as in archaebacteria, which is not shown here. For example, in mitochondria, the stop codon AGA, AGG, which are encoding arginine, sense codon, but in mitochondrial system they are stop codon. In addition, UGA and UAG, although they are stop codon, but they also specify the newly discovered amino acid. So because of these differences, genetic codes is considered as almost universal, not strictly universal. The next property of genetic code is codon bias. Although the genetic code is degenerate, having multiple codons for the same amino acid, these synonymous codons are not used with equal frequency or preference. This is due to differences in the abundance and availability of their corresponding tRNA. It means that some genetic code in a group is preferred over the other. This is known as codon bias. For example, there are about 8 genetic code. Bacteria cannot read these because there is no tRNA available in the cell. Is it bad for the bacteria or it is good for the bacteria? Basically, it is normal for bacteria. It will not affect the survival. But it has technical implications. If you are trying to express your gene in bacteria, it might have technical implications, which I will explain in the next slide. So what are technical implications of codon bias? If you are planning to express certain eukaryotic DNA fragments which are rich in cytosine, you may have problem in their expression in bacterial host because you are likely to hit one of those 8 genetic codes which are not read by bacteria. So how you will solve this problem? We will explain that in the next slide. So you can solve this problem by using specially designed recombinant bacterial host cells. For example, RIL stain, which has tRNA for arginine, isoleucine and leucine, or alternatively you can use Rosetta stain, which has tRNA for proline and arginine. So using these commercially available recombinant stain, you can solve your problem. Thank you very much for watching this clip. I hope this will help you to understand the concept behind genetic code. Please like it, share it, and subscribe it. Until next time, you take care. Bye bye.